Is that right? Okay. Again, huge apologies for the delay in joining, but I'm glad that we're here. So today I'll talk about some emerging work that we're doing in both the preclinical and clinical setting in the context of specifically behavioral neurodevelopmental disorder known as autism, autism spectrum disorder that we're all familiar with, and highlight some of the basic research that has led to a small early stage clinical trial that shows interesting and potentially promising evidence that we may be able to intervene in autism at the level of the gut. And of course, for KAT6, there's GI implications, and so perhaps some of the discoveries we've made in the autism space may be translatable to other disorders. And so just a little bit of background on the microbiome. So we are teeming with bacteria on all environmentally exposed surfaces of our bodies. About 100 trillion bacteria occupy the ecosystem that we provide to microbes. Most of these organisms are in our lower gastrointestinal tract, specifically the colon. There are more microbial cells in our bodies than there are human cells, and 150 times as many genes in our microbiome, which is a collective genomes of our gut bacteria, than in our own genome. And really this concept that the microbiome is a forgotten or an alien organ is reinforced by evidence that the microbiome itself has a metabolic capacity to the liver and actually a similar mass to the adult brain. And so, as I mentioned, the focus of my talk today will be on autism spectrum disorder, but a number of different neurological conditions have been linked to the microbiome. Most of this work is in preclinical models, so I just want to caution that there's still a long way to go between knowing whether the results that have been identified by many labs around the world in mouse models of behavior or cognition or even motor deficits translate to humans. So again, it's a large unknown. But in addition to autism, as you can see, anxiety and depression, cognitive issues, including Alzheimer's and motor deficits, neurodegeneration, such as Parkinson's, have been linked to the microbiome. A number of metabolic disorders, such as diabetes and cardiovascular disease and obesity. So a lot of research, again, in preclinical models and some early stage clinical work in obesity that have linked that condition to the microbiome. And of course, immune-related disorders, whether autoimmunity or inflammation or infections, also seem to be impacted by the composition of gut bacteria. So speaking specifically about the gut-brain connection, there's a number of different conduits or pathways by which the gut and the brain can communicate. So in this schematic, what you see here is, just essentially a cartoon of what would be the gut, and this would be, let's say, the systemic compartment, and then the brain, highlighting, that there are small molecules produced in the gut under regulation by the microbiome, such as serotonin, 5-HT, and a number of other hormones and neurotransmitters that, again, are produced by human cells or mouse cells, directed by the microbiome, that then can impact neurological activity or behaviors. Certainly a role for the immune system in all of these neurodevelopmental conditions. Many up here, or all of them, seem to have some immune component, whether it's a driver of neurological outcomes, a facilitator, or a bystander, consequence. I think that's a question that still is open, but certainly you see immune dysregulation in many different types of neurological and neurodevelopmental conditions. And then the immune system, again, these immune cells can traffic from the gut to the brain. About 70% of our immune cells are in our intestines at any one given point in time. They can traffic to the brain or to adjacent regions of the brain, such as the meninges, and then send their signals into modulating neuronal function. What I'll talk about today is this pathway by which small molecules produced by bacteria, so again, origins of bacterial metabolism, can transfer through the gut and then into the circulation. And then there's obviously neuronal connections, where they're the vagus nerve that transmits in both directions, or adrenergic signaling from the brain or the spinal cord that can innervate the gut. So certainly there are neuronal conduits as well. So again, as I mentioned, in the context of our work, we focus on autism spectrum disorder, And as you likely know, autism is diagnosed by behavioral assessments. There are no reliable brain scans or blood tests to diagnose autism or to assess its severity. But we do, again, have essentially questionnaires, both physician or behavioral therapist administer questionnaires or parental questionnaires or both, that look at the domains of social communication and repetitive behaviors. Again, these are the core features of autism. But many years ago, what caught our eye in thinking about non-behavioral manifestations of autism are, whether it's anxiety or hyperactivity or seizures, certainly are part of large populations of the autism community. Not all of them, but many of them will suffer from these comorbidities. But immune dysregulation, metabolic abnormalities, which again, as I highlighted, are influenced by the microbiome, and very curiously, gastrointestinal symptoms, depending on the study or depending on the type of symptom, about 50%, ranges between 30 and 60% of individuals with autism will have some form of gastrointestinal symptom. I'll come back to this in a minute. And so many years ago, we started working with Paul Patterson, who's pictured here. Paul was a behavioral neuroscientist at Caltech, and he was very interested in the role of the environment in modulating complex behaviors. Paul unfortunately passed away in 2014. But he had developed this mouse model called the maternal immune activation model, and it was based on epidemiologic data that showed that an infection during pregnancy leads to increased risk of autism in a number of different cohorts, European cohorts, as well as more recently cohorts here in the US. And so what he did is he modeled this epidemiologic data by essentially giving pregnant mice the flu, and then later used a chemical called poly-IC, which is a viral mimic, similar to, it essentially activates the immune system similar to what a flu would, and then showed that these animals had behavioral abnormalities very consistent with autism. Later, he and others showed that there are abnormalities consistent with schizophrenia in this model as well, which actually shares that epidemiology of maternal infection and maternal immune activation that shares that with autism. And then certain neuropathologies in these animals, which are reminiscent, these are not hallmarks of autism, but have been shown in certain postmortem studies to be correlated or increased in individuals with autism. And so these mice show these behavioral effects as well. And so when we started working with Paul, and this 2013 paper was the first publication for our collaboration, we asked, and I'm going to skip a little bit of data just for the because of time constraints. What we asked in this study is, are there GI issues in these animals? And the answer was yes. Again, I refer you to this 2013 paper. These mice had GI issues, again, similar to what's been shown in human autism. They had microbiome changes. There's a number of studies which have shown the microbiome is different in individuals with autism compared to neurotypical or matched controls. And we showed that to be effective as well. And then we gave these animals a probiotic called Bacteroides fragilis, here abbreviated as BF, and showed that Bacteroides fragilis was able to ameliorate many of their behavioral symptoms in the MIA model. And then so again, in the same study, towards the end of the study, we started wondering, what is that communication or the connection between the gut and the brain? And so again, this is why I alluded to that in a few slides ago, about the fact that the gut and the brain have this rich, essentially, communication that's bidirectional, goes in both directions. And so we profiled the immune system, we published that data. It didn't seem to have the largest effect. We haven't yet looked at neuronal signaling, but in this study, we looked at microbial metabolites, or just small molecules in general that are different between these groups of mice. And so S is the control animal. So these animals, their mothers were given saline. These animals, their mothers were given poly-IC, which is again that viral mimic that activates the immune system similar to the flu. And you can see that there's this metabolite called 4-ethylphenyl sulfate, or 4-EPS, which is quite dramatically elevated in the poly-IC offspring. Again, these are the mice that have the behavioral abnormalities and the neuropathologies that are consistent with autism. And we see this pretty large increase in this one molecule. And then this metabolite is then reduced to near normal levels in the animals, the polycy animals that were given the probiotic. Again, the probiotic is not the focus of the talk today, but it was a research tool that led us to this metabolite. And 4th-phenyl sulfate had really not been studied. There was only one paper in the literature which showed its chemical synthesis. It had been predicted to be a product of microbial metabolism. It doesn't come from food. To the best of our knowledge, we don't make this. Mice, even flies and worms don't appear to make 4th-phenyl sulfate. It appears to come from bacteria. And we've surmised that there was a biochemical pathway that converted tyrosine to 4-ethylphenyl. And then once 4-ethylphenyl was produced by the bacteria, was sulfated to 4-ethylphenyl sulfate in the liver. And ultimately, that's why we detect 4-ethylphenyl sulfate in the circulation, is because there's a pretty quantitative conversion. And this sulfation mechanism is essentially a detoxifying mechanism for the metabolite. And this is similar to several other metabolites, which had been shown to be urinary metabolites associated with autism. So we thought maybe this is more than a coincidence that the one molecule so highly elevated in our data set is quite similar, one carbon different from paracresol sulfate and similar in structure to indoxyl sulfate. Again, these metabolites had been shown to be elevated in human autism. As I mentioned, 4-EPS had not been previously studied. And so we set out first to ask, can we validate the mouse data in humans? And so we worked with Paul Ashwood and others at UC Davis, and we were able to acquire samples from the CHARGE study, which was a very large NIH-funded study that collected plasma and a lot of data on a large cohort of individuals with autism, with ASD, and then typically developing controls. But they, I think they were very wise, in my personal opinion, but also, we're able to see a little bit into the future. And we're 10 years ago collecting feces in an autism study, which again, was quite atypical to be collecting that tissue. But again, we benefited from this, and we were fortunate to be able to get these samples and then perform high throughput metabolomics, and indeed showed that in the plasma, there's an elevation of 4-phenyl sulfate in a subset of individuals with autism. So you see about 1/3 of the ASD population have really high levels of this metabolite compared to those in the typically developing control. There's a group here that have these higher levels that are typically developing. We don't know much about the typically developing population controls. They were not behaviorally tested. And so it turns out that the individuals that had higher levels of 4-phenyl sulfate, they seem to have the worst social behaviors of the group. And then those individuals that also had GI issues had the highest level, 9-fold over typically developing. So a lot of what we had discovered in mice appears to be at least correlated in humans. That gave us, that emboldened us to think about a research program where we went out and we asked, does 4-ethylphenyl sulfate itself have a biological function? Meaning, just because we detected it in coincidence with behavioral alterations in mice and with a diagnosis of autism, it doesn't mean that it's part of the pathophysiology of disease. It could just be associated. It could be a biomarker, which I think would be interesting in and of itself, but it doesn't mean it's functional, just an association. And so we wanted to go from correlation to causation. And so we worked with Michael Fischbach at Stanford to essentially discover what is that microbial pathway. And we others have proposed that this metabolite is of microbial origin. And so Michael, using both computational and experimental approaches, identified the 3 genes, bacterial genes, required to convert tyrosine, which is found in the gut, and again, is just an amino acid that we consume in our diet. That tyrosine is converted to parecomaric acid, to 4-vinylphenol, and then to 4-ethylphenol. This is what the bacteria get produced in the gut. As soon as they secrete it, it enters the circulation, and then very quickly, as I've already mentioned, is sulfated in the liver by this mouse and human enzyme called SALT1A1. And so once we had uncovered the pathway, we were able to engineer bacteria that either did not produce or did produce 4-ethylphenol. Same exact bacterial strains, just either they contain the 3 genes or they do not contain the 3 genes. And so again, a cognate set of bacterial organisms that are different in one metabolic pathway, and that's the production of 4-ethylphenol. And so we took these bacteria and then we colonized then 2 groups of mice. So these mice got the bacteria that were bioengineered to not produce 4-ethylphenol. These mice got bacteria that did produce 4-ethylphenol. And so again, now we have a very clean system. It's a very reductionist system. It's not physiologic, but we have a very clean system where we have two groups of mice that, again, in their entire bodies only differ by this one metabolic pathway, right? So we can ask what are the consequences of mice that are either exposed to 4th-phenyl in the gut or not. And so first we want to just validate, indeed, the mice that receive the bacteria that are bioengineered to produce 4th-phenyl, they indeed do so. We see 4th-phenyl in the feces, we see 4th-phenyl sulfate in the urine, this is in blood as well. And then when we looked in the brains of these animals, a small percentage of that circulatory 4th-phenyl sulfate does enter the brain. So it's suggestive, it doesn't prove that it's directly acting on brain cells, but it's suggestive that it may have a function. So then we work with Daniel Holschneider at USC, University of Southern California, to ask, are there changes at a global level in brain function and brain activity? So there's a 2-deoxyglucose uptake experiment where radioactive glucose was given to the animals before a behavior test and an open field test, essentially just activates different brain regions. And then the tracer, the 2-deoxyglucose, is incorporated into those brain regions that are more active, and then incorporated less than baseline in regions that are less active. And so what this gives us essentially a map of the brains of these animals that are more active in the 4EP group, and that's shown in red, and then less active in the 4EP group, shown in blue, compared to the 4EP negative mice. So what's really interesting is that a number of these brain regions are part of the limbic system, which controls fear and anxiety and other emotions, which makes a lot of sense considering the association to autism and neurodevelopment. And it also hinted to the fact that there may be connectivity issues. That's not simply that different brain regions are more or less activated, but that different brain regions are not talking to each other, not communicating to each other in ways that, let's say, mice that weren't exposed to 4th-phenyl would exhibit. In fact, I don't have the time to do so, but we did a functional ultrasound very similar to a functional MRI to show that indeed there are connectivity changes. And that's important because similar changes have been shown, changes in structural and functional connectivity have been shown in the autism, in individuals with autism. And so after that global change in brain activity, we want to drill down to the cellular and molecular level and ask, what is 4EP doing to these animals that leads to these brain-wide changes? And so we worked with Dan Greshwind at UCLA to perform an RNA-seq experiment where we looked at 6 different brain regions. These are all the brain regions that were informed by that previous experiment that I showed you that were either more or less activated. And then gene expression by RNA-seq gives us, again, molecular insight into what are changes in particular cells in the brain. And the vast majority of the changes were in a region called the paraventricular nucleus of the thalamus. I can just highlight that region here. You can see it's more activated in the 4EP positive group. And then what this pie chart shows are differentially expressed genes. So in the entire data set, where are most of the changes? 6 brain regions, the vast majority, over 3/4 of the changes, are in this one region, the PVT. So it really highlights that region as maybe an area of activity that is associated with exposure to 4-ethylphenyl sulfate. And then when we did a cell enrichment analysis, we saw there are changes in neurons. We're following up on these changes now, but a lot of changes in this cell types called oligodendrocytes. And I'll introduce these cells to you in a minute. I'll introduce them to you actually now. Oligodendrocytes are cells that modulate myelination patterns in the brain. They essentially coat neurons with a protective shield, if you will, that allows them to transmit electrical impulses over long regions, similar to a coating on a copper wire in your house. And so the gene expression studies suggest there are changes in oligodendrocytes. So we went down and we looked at those specific genes which mark different populations of oligodendrocytes. And so OPCs are oligodendrocyte precursor cells. These are immature oligodendrocytes. And you can see in the 4EP positive group, so these are, again, different genes. I'll note it here, comparing 4EP negative and 4EP positive. All of the genes that are hallmarks of immature oligodendrocyte precursor cells are elevated in the 4EP positive group, suggesting that 4EP exposure leads to an arrest in oligodendrocyte maturation. And gratifyingly, we see the cognate effect on mature oligodendrocytes. And you see all the genes that are associated with mature oligodendrocytes are reduced in the 4EP positive group. So a really strong signal that indeed what we're seeing in the brain are changes in or essentially are some sort of a block or a delay in the maturation of oligodendrocytes. So as I mentioned, oligodendrocytes, they can mature from this precursor state into these myelinating oligodendrocytes. And again, their primary function is to myelinate neurons. And so what I showed you previously is a gene expression pattern. So we went into the brains of these animals. We actually looked for these cells and phenotyped their maturation state. And indeed, the oligodendrocytes in the brains of 4EP positive mice are less mature than the oligodendrocytes in the brains of the 4EP negative group. And this is by looking at cell surface markers that are associated with oligodendrocytes, initially done by immunohistochemistry. We repeated these studies with flow cytometry. So here we're dissociating the brains of these animals and then running the samples through flow cytometry so we can look at an individual cell basis for expression of mature or immature oligodendrocyte markers. And indeed, we see the exact same pattern. So very gratifying to see both in the brains and ex vivo that there are changes in oligodendrocytes. And it appears that 4EP, again, arrests oligodendrocyte maturation. And as I mentioned, the function of oligodendrocytes, at least the primary function, is to myelinate neurons. So in the next step in the project was to immediately, or directly look at oligodendrocyte or look at myelination patterns in the brain. And so here we're using electron tomography. So there's no label here. It's not, we don't use an antibody or any other indirect label. We're looking directly imaging neurons, but electron tomography gives us the resolution to actually look at how thick and how organized the myelin is. And indeed, we see more unmyelinated axons in the 4EP positive group and a higher G ratio, which takes into account cell shape irregularities. A higher G ratio is associated with thinner myelin. And if we actually look at the thickness of the myelin directly, these are myelin sheets here, we see that the myelin is thinner in the 4EP positive versus the 4EP negative group. So all the sort of the hypothesis seems to hold that the gene expression changes in the brain, which lead to oligodendrocyte changes, directly lead to changes in myelination. And again, myelination changes have been shown in the autistic brain, as well as in animal models of autism. And of course, we want to bring this back to behavior to try and answer that central question of whether or not this metabolite was actively involved in modulating behavior. And so we ran a couple of behavioral tests. So this is the open field test that measures anxiety, and mice that were exposed to 4EP enter the center less than the 4EP negative group. So they're more anxious because the more bold the mouse is, the more it'll enter the center. The more anxious the mouse is, the more it'll stay towards the walls. And indeed, these mice stay towards the walls. And if we use the elevated plus maze, we put the mouse on a platform that's about 4 feet off the ground. There are closed arms and open arms. Anxious mice stay more in the closed arms. Bold mice will enter into the open arms, and we measure how much time the mice spend at the terminus of these open arms in the 4EP positive group, spend less time at the terminus of the open arms in the 4EP negative group. So these mice really have anxiety. We also show that they have social deficits and vocalization changes as well. I didn't have time to show that. Those are more autism-related behaviors. And to summarize the preclinical data, we show that changes in the microbiome lead to changes in metabolites such as 4EP. These metabolites can cross the epithelial barrier, get into the circulation. They're modified by the host. So this is really co-metabolism by the microbe and its host into a final product as 4-ethylphenyl sulfate. As those concentrations increase, some small proportion of 4-EPS enters the brain, and then that leads to an arrest in oligodendrocyte maturation, changes in myelination patterns, and altered emotional behaviors. And so really, this preclinical work gave us confidence to think about ways of translating these findings into humans. And we took a number of different approaches, at least we thought and planned about a number of different approaches of how we may utilize this information to develop a drug. And what we settled on was an approach where if the hypothesis is that an elevation of these neuroactive, maybe even neurotoxic molecules in the gut is associated with behavioral changes, the mouse studies suggest that they're actually causing the behavioral changes. And what if we just reduce the levels of these microbial metabolites? And again, I showed you from that human study that these are metabolites that are elevated in the plasma. I didn't show you the feces data, but they're elevated in the feces of individuals with autism as well. So the approach that we took was to give a drug or to develop a drug that was a sequestrant. It is able to bind this class of small molecules with high affinity. And importantly, the drug is non-absorbable, meaning it's taken orally, but it does not get into the circulation. So the drug only stays in the gut. And the hypothesis is that as this drug is moving through the gut, it is binding these small molecules of this chemical class and then excreting, and once it's bound to the drug, then it's excreted with the drug, effectively lowering the concentration of the metabolite in the drug, and then by association, lowering the level of these metabolites in the circulation. In fact, we validated that in mice. But what's most, I think, striking is that this drug was able to improve behaviors in the mouse model. So these are the data I showed you for the open field test, where the 4AP positive mice spend less time in the open field. This phenotype was corrected when we gave those same animals the drug, right? Same in the elevated plus mice spend less time in the open arm of the arena, more time if they were given the therapeutic drug. I didn't show you previously the marble bearing test. This is a test for repetitive behavior and for anxiety. And the 4AP positive mice have elevated levels of marble bearing. Again, this anxiety-like phenotype that's corrected with the drug as well. And so with this data, we went into a small clinical trial where, again, open to remember, this was an open label trial. Everybody in the trial got the drug. So we don't have a placebo control. So please bear that in mind. But the design of the study was a 1-month screening where individuals who had a prior diagnosis of autism and self-reported GI issues, gastrointestinal issues, were brought into the clinic. They were screened both for behavioral tests and for GI tests, and then samples were taken. And individuals who, again, they were previously reported to have autism, we validated that with our own behavioral instruments. Again, not our own, meaning that we administer these behavioral instruments. We did not develop any of these behavioral tests. But we validated that individuals had autism, again, after the screening period. And then the therapeutic dosing was 1 week on low dose, 2 weeks on low dose, 2 weeks on a medium dose, and then 4 weeks on a high dose. You can see these green triangles are where they came in for office visits for behavioral testing, and for sample collection at the end of trial, which is here in blue, as well as behavioral testing and collection of samples. And importantly, we did a 1-month washout. So a follow-up after 4 weeks, they came back in for additional behavior testing and metabolomics. And again, here are the demographics of the individuals in the trial. The study was done in Australia and New Zealand. And so it's a safety and tolerability trial, and it was a target engagement trial. It was not powered to show efficacy. And so there were no adverse events. This is all in this paper that came out a few months ago. The drug was very, very well tolerated. And in terms of target engagement, this is for phenol sulfate. I mentioned paraquisol sulfate. I mentioned doxyl sulfate. This is, again, a class of phenolic molecules produced by gut bacteria. And we were able to lower circulatory concentrations in humans with this drug. So you're looking at plasma levels at the baseline. So this is visit 2, and then end of trial, visit 5. So it's a comparison of the baseline and then the end of trial, again, separated by 8 weeks of drug. And you can see that all of the metabolites that are associated with this chemical class are reduced in the circulation in those people after 8 weeks on drug. And then there's a control metabolite, which its structure suggested it would not be affected by the oral sequestration, and indeed it wasn't. So this is essentially a control that we're not depleting everything, but we're depleting those chemicals that we had shown are that have a high, or the drug that for which the drug has a high affinity. So again, we're very, very happy about the target engagement. But we were also fortunate, again, these are preliminary data, but we were also fortunate to see a statistically significant improvement in behavior. Again, the study was not powered for behavior, but the effects, at least for anxiety and irritability, were strong enough that even with 26 participants, we were able to see significant improvement. And so using the pediatric anxiety rating scale, these are all participants, again, baseline, end of trial, and you see a decrease in their PAR scores. A PAR score of 10 is considered by some to be clinically relevant. So we took the cutoff of those individuals who came in with anxiety. So again, not every person with an autism diagnosis will have an anxiety diagnosis based on the PARs, but those who did, we looked at them, and over the 8-week treatment period, most of them improved in their anxiety. You see this is quite significant. And then also with irritability, this is the entire trial. It was significant for all participants, but when we take a cutoff of 15 for the autism behavioral checklist, this is subscale I for irritability. If we take all those individuals who had irritability coming into the trial, you see that the vast majority of them, again, improved in the 8 weeks on drug. And again, this is not placebo controlled, so we take all this data with a grain of salt, but I think the signals, or I know the signals here are quite strong. We can compare our placebo effect or our presumed placebo effect to previous trials in autism, and it looks like even if we were to calculate a placebo effect that's consistent with other trials that were designed very, very similarly, we have a very, very strong signal. The only way we'll know that is with the phase 2 trial that is now ongoing, that is a randomized placebo-controlled trial. And so I'll just leave with this concept that current approaches pharmacologically try to get drugs into the brain. Our work considers the role of the gut-brain connection. And if there's this dialogue between the gut and the brain, and part of that dialogue is mediated by microbes and their molecules, then one can intervene at the level of the gut and potentially get improvements in the brain. Again, this is what the drug that I just showed does, that it's, again, an oral drug that is not absorbed, so it only works in the gut, but it really impacts the signal, right, that the gut is sending to the brain. And the advantages here, I think, are obvious, is that getting drugs across the blood-brain barrier, the BBB, is very challenging to get pharmacological doses in the right brain regions. There's a lot of off-target and on-target effects that are undesirable. But if the drug is only in the gut, it's likely to be safer, it's likely to have less off-target effects, and again, leverages the biology that was forged by the gut-brain connection. So again, this is a hypothesis, it's a concept, partially being tested by the current trial, and maybe I'll give you the link to those who are interested. It's theautismstudy.com. Theautismstudy.com is the website you can go to if you're interested in the trial. So I'll end and thank the people who did the work. So Brittany Needham was a postdoc in our laboratory. She just started this month her own laboratory at Indiana University as a tenure-track professor. Mark Wei Lee, former lab members who assisted Brittany in her project. Our work is highly collaborative. I think I've mentioned many, but not all of our collaborators, both in academia and in industry. So Axial Therapeutics ran the trial that I mentioned, and these are our funding sources. So sorry for the technical issues, and I'm happy to take questions.
Thank you, Dr. Mazmanian, for a powerful presentation. What a fascinating relationship between the gut bacteria and brain health. So the webinar is open for questions, and please feel free to also pop your questions in the chat function. I see Dr. Ann Watts has a question.
Yes, thank you very much for your very interesting talk. I wanted to know, with the predominant anxiety improvement in the animal model, is there any reason why you wouldn't also assess people who just have anxiety together with a gastrointestinal complaint?
Very good question, and we started that research. We've started it with the approach of looking for these metabolites in individuals with high trait anxiety or major depressive disorder. In humans, I think, again, the mouse models, even though we identified 4 EPS in mice, I think we all agree that it's better to start with human data and then work backwards to see if we see an effect, to see if we can model that in animals. And we're getting some preliminary data. It's still very early. We're getting some preliminary data to suggest that indeed there are elevations of 4-EPS and other related metabolites in individuals with anxiety and depression. We're now just powering those studies and getting those samples up to hopefully reach significance. But that's really, again, emboldened us to think about, to your point, going beyond autism, right? At this point, I don't know if this is an autism-specific phenomenon or a more generalizable phenomenon that may even relate to other disorders. And these are the questions that we're trying to answer. But initially, it looks like there may be, at least a glimpse that individuals without autism, but with an anxiety or depression diagnosis may have a similar chemical profile.
Maybe if I can ask one short other question. I joined a little bit late, and you may have said that, but how common are gastrointestinal complaints in association with autism? What percentage of patients would have that?
Yeah, roughly 50%. If you look across a number of different studies, but of course, there's a range. And then the types of symptom manifestations are abdominal cramps, bloating, abdominal pain when that can be communicated by the subject, diarrhea, constipation, foul-smelling stools, changes in consistency. But there doesn't seem, and I go through this list because there doesn't seem to be any one type of GI issue. And it appears that individuals who have gastrointestinal symptoms in the autism community have gastrointestinal symptoms, even for them, it changes over time, that they may have, bloating and constipation as a major, complaint, and then sometime later, diarrhea is a major complaint. And so again, I would say we don't really have we have some like rough numbers of who reports GI symptoms. I don't think we have a good idea of what those symptoms are. We certainly don't know what is causing or changing those GI symptoms.
Thank you very much.
All right, I'm going to take some questions from the chat. So Dr. Matthew Todd, great talk and great results. Has modified diet ever been tried on the mouse model? Restriction of phenolic amino acids in the diet.
We considered this, but since tyrosine is so important, and there's other amino acids such as tryptophan, which can give rise to similar molecules, not 4-EPS, but similar molecules. For example, indoxasulfate comes from tryptophan. We haven't thought specifically about diets, though I suspect it's not our area of research or certainly not my area of expertise. I suspect there may be ways of modifying diet to change these molecules. And as I mentioned earlier, we did think about a number of different ways to intervene. Diet was one of them. But I think there were other reasons, certainly from a clinical standpoint. What I've been told, and I think maybe this is intuitive as well, is that dietary interventions often have a nutrition rate that's higher than people who are taking medication. So the answer is unfortunately no, though I do think there may be viable ways to approach this issue by modifying diet. At the end of the day, and I think, again, it's intuitive, at the end of the day, the molecules that the microbes are metabolizing, what they're producing, are almost entirely the product of dietary substrates. What we eat, what we ingest, they have first crack at it, if you will, right? And they metabolize these molecules, they change them, they transform into many different compounds, but the substrates come from diet. So it makes sense, again, that dietary interventions may someday be useful.
Okay, I'm going to take one more question, and this is from a parent. Great information. Any opportunities for us to have our children participate in similar trial in the future?
I'll put it into the chat because I just mentioned it.
Okay.
Autism.
And I have one question, Dr. Mazmanian. I don't have a genetics background or microbiology background, but I do want to understand how to measure the microbiome, like qualitatively and quantitatively. Like, what are the resources that are used to measure it?
Yeah, so we generally use an approach called metagenomics these days, which allows us to essentially sequence all of the DNA in the microbiome. That gives you both an image of what are the organisms that are there, because then once you have the sequences, you can assemble their genomes and assign it to bacterial species, a genus and a species. Most of the time, I think we're getting better at making those assignments. 5-10 years ago, there were a lot of bacteria that were unassigned. right? So we can essentially get a catalog of organisms. But what a metagenomics allows us to do is to get a catalog of the genes, like not just what, it's like the name of the species, of the organism, the genus and the species, but what genes are harbored in its genome, in its DNA, because again, that's what's going to produce RNA or protein that's going to have a biological function. Metagenomics allows us to essentially make predictions towards function of a microbiome. But again, it's a pretty well-advanced technology these days. There are companies that do this as well as academic labs. And so I think the concept, the ability, and there are probably, for your audience, service providers that you can find where you can send your stool sample just as a, anyone in the public can send their stool sample and have it analyzed by metagenomics, and you can get back the data. I caution you, you're not going to be able to interpret most of the data that you get, and not sure like what the actions are. But I think these days, like you can go to americangut.com, or sorry, americangut.org, and for $100 get your microbiome sequence.
Okay, all right, thank you. And I have one more question. So you talked about the 4 EPS and its elevation in the plasma. You also talked about the feces that were collected in the mouse model. So what are the biomarkers? Like how is that information processed? Can you tell us a little bit about that?
Is your question, in terms of a biomarker question, or is there a question in terms of what is produced where, and how is it, how are the two chemicals related?
Yes, what is produced where and how is it related?
So the bacteria, again, the bacteria live only in the gut. So there's no evidence, at least to our knowledge in mice or in humans, that in autism and many other conditions, that bacteria actually translocate or leave the gut, right? So the source of the chemicals, is almost certainly in the gut, right? Then the bacteria are not in our circulation producing these molecules. And again, what they produce in the intestine is 4-ethylphenol. Right? Okay. Once 4-ethylphenol gets across the epithelial barrier and into the circulation, is very quickly sulfated in the liver to 4-EPS. And as I mentioned, we only see 4-EPS in the circulation of mice. We don't see 4-EP. We don't even see 4-EP in humans. We only see 4-EPS. It's a very quantitative, very robust conversion. And it's known why we do this, right? And it's been studied in like chronic kidney disease and other disorders of why we sulfate small molecules. Many of these phenolic compounds tend to be toxic, right? Toxic largely to the liver, right? And they have some other, sort of toxicities to other tissues. And so once our bodies are exposed to these molecules, we sulfate them so that we can excrete them in our urine. That's why you actually pick up many of these metabolites in the urine.
Okay.
And the urinary biomarkers, because our body's trying to get rid of them by sulfating them.
Right. Okay. All right. Thank you so much for your time. And the terms like gut feeling, gut wrenching, they make so much sense to me now. So thanks a lot. So let's move on to the next presentation. Back in 2020,